Hey, apologies, I'm late. We'll start in a bit. My computer crashed, so I have to start everything up separately. Old school, eh? So let's see, where is this screen? Here we are. So again, if you have any questions, you can ask me in the chat and I will try to keep an eye on that. Okay, yeah, sorry for the delay. I was in a meeting, it took too long, and here we are. Okay, apologies for the delay. Let's start, because there is so much awesomeness still to discover. Yeah, exactly. Who is that? Bonus points, yeah. Yes, because we've now, so it's now inside the data folder for the files. So you can find a prf_Lyon tar.gz file in that files data folder. And similarly, you can find a reports, I believe, .tar.gz file in that same folder. The reports is relatively hefty at 2.4 gigabytes, but you know. So I'm using these again. I hope this works. Let me check. AirPods Pro, yeah, why not? Can you still hear me? Anxiously waiting. Yes, excellent. Okay, so to highlight where we are, right? Because I understand that you know, the course is somewhat random seeming in terms of its structure. I apologize for that. But I just want to recap where we are. I think so this is actually the final lecture for all previous versions of this course. That is to say, in earlier years, we had one theory lecture per week, And of course, last 8 weeks. We're now in the 4th week, so this would have been near the end. But we're compressing this theory part because we want to put you all to work with these projects. Right? So this week is the transition from the end of theory to the projects, and Friday I will explain to you what the projects are about, how they work, and give you a bit of introduction, right? So come Friday, we'll be discussing the content of the projects, but also the logistics of the projects. That is the groups that you will form to do the projects. And I'll try to make it very clear to you what extra parts of the literature would be good to read, having chosen one of the projects. Yes? So that's where we are. And I think this makes sense because we've now gone through the basics of fMRI analysis. We've gone through pre-processing. You have an idea of the things that you can do with fMRI data, right? The things that need to be done, but also the things that you can do. You can do a basic GLM analysis. You know where the blobs in brains came from. We've also talked about these patterns of brain activity that are interesting, or can be interesting, right? Why they're interesting and why they may not be interesting. And we've also talked about the value of looking at that single voxel that we're recording from. And I've tried to convince you of the idea that the better we can understand the single unit of measurement, in our case, the voxel, the more we understand about what the brain is doing computationally, right? If we like to say our brain is a computer, then we should be able to find out what the program is that computer is running, right? And so I think that this single voxel modeling thing gives us a very clear idea of what the brain could be doing, and that's really where the field is going. But today, I want to sort of close off that theory part because there's one thing that we haven't talked much about, and that is the idea of connectivity. That is, if you are measuring the brain over time, not only can you relate those brain activations to what happened in your experiment, and for example, do modeling of single time courses or find blobs, you can also look at how the brain fluctuations that you see in your data, how they co-vary. Yeah, and so that's what we'll be talking about today, this notion of connectivity. It may seem a bit vague now, but I hope at the end it will be less vague. So connectivity, what does that mean? So it's a really broad scale idea. And so this is sort of a hypey thing. Also, people think of the wiring of the brain in its totality. So that is all of the connections between all of your nerve cells in the brain. People have called that the connectome. Right? The way that we have a genome, which is the totality of our genes, we would also have a totality of connections in our heads, which would be the connectome. And then, of course, this computer that our brains are, they would be defined by this connectome. Sounds really cool, right? And so what we need to do is, map out all of these connections, map out the connectome, and we would have a very, very clear idea of what the brain is doing. So that's what this relates to. Seeing the brain not as things that happen in terms of patterns, not as things that happen at every single voxel, but really trying to see the brain as this network. Yeah. I'm still in Zoom, apparently. Who knew? So, we've now done this thing where we can analyze experiments that have a task, right? We can do things like pre-processing, analyze these with a GLM. And we've more or less been focusing on single voxels in a parallel way. Yeah? So in the notebooks that you've been looking at, we're really looking at single voxels all the time. And then the idea is that, you can do this analysis on every voxel in parallel, and that gives you results. Yeah? So it's important to realize that when you're doing this, you're
you're in a model-based sense, referring bold signals to a design matrix, right? You're drawing a relation between what happened in the experiment and every single voxel. And this gives you blobs, or this can give you model-based fits, like in this single voxel modeling case. But whatever happens in your brain doesn't just depend on the on what's happening on the screen, for example, or the reaction time of your subject in your experiment, but it is also contingent on what's happening in other brain regions. Yeah, it's not you're not just a single reflection of what's happening on a screen, but there is internal processing that will differ, for example, from trial to trial. Right, so that brain state, that sort of situation that you're in mentally, that isn't just a function of the stimulus. It is actually also a function of what is happening in the brain as a whole. Yeah, and this is much more complicated, right? You have to think of the brain not just as a loose collection of voxels, which we have been doing up until now. There might be patterns across this collection of voxels, right? But what we're interested in now is really the sort of state of this processing machine that the brain is in, right? And there, it matters what every voxel is doing at this moment in time. Right? We're really interested in the instantaneous pattern of activation across all voxels. Is it clear that this is different from the notion of the pattern analysis that we've spoken about before? Right? Patterns can be average, for example, across many trials. What we're talking about here is really how things fluctuate, and that one trial can be completely different from another trial, even if the stimulus is the same, because there are different brain activations. Yeah. Okay, so what I want to do is first zoom into it and try to give you a bit of background and talk a bit about parcellation of the brain, the different methods that we have, and then things will become clearer. What do we mean with this connectivity? So we can distinguish two types of connectivity. So the first is that we can look at anatomical measures that relate to connectivity, and they can be seen as sort of anatomical backbone. We've talked a bit about DTI, but you can see this as the backbone of the connections in the brain, and I'll get into this in a bit. This anatomical sort of fixed relations to connections. But we can also do functional imaging of connectivity. Yeah. So my goal is that we'll go into this functional connectivity towards the end of the lecture. Now, this has to do with the fact, of course, that the separation between anatomical and functional connectivity has to do with the fact that we can use the scanner in different ways. Right? So we can do this DTI imaging to get at the anatomical connections, for example. We get the structural brain network, but we can also perform time series analyses. Right? So this is fMRI, and this is anatomical MRI. But what you see here, this anatomical parcellation, is something that I'm going to go into now. So I first want to focus on this left side, the fixed connections in the brain, and how can we make sense of them. And so this anatomical connectivity, because of course, if you don't have a physical connection between brain regions, you're much less likely to have a functional connection also between brain regions. Yeah. So this parcellation that I already dropped as a word. Normally what we do is we can, what we want is to designate regions of interest. We've talked about that before, right? You want to select a specific portion of the brain and analyze signals from that portion of the brain. But how do you decide what the boundaries of each of these patches are in the brain? How do you discern one region of interest from the next? And so we can do this based on a lot of different types of information. We can take anatomical images from the MRI scanner and do things like myelin. So we can look at the histology of the tissue in the brain and look, for example, at how much myelin is in the gray matter. We can also do this based on functional imaging. I've shown you examples of these retinotopic maps, for example. We can draw boundaries between the retinotopic maps in the brain, and that gives you a parcellation based on functional imaging. You can also show people faces and show people houses, and then you get a blob, and you can draw a circle around any one of these blobs. And this also gives you a functional parcellation of the brain. You can also just say, this is part of the superior frontal lobe, right? So you base this on an atlas of anatomy. So you co-register a subject with some atlas and thereby just say, this is this region of the brain. And all these types of information can come together to designate what constitutes a region in the brain. And then what you can also do in terms of anatomical connectivity is this direct assessment of communication tracts by means of tractography. I think I showed you an example of this earlier on. It's DTI. Now, you can do this parcellation based on histology in many, many different ways. So here I'm showing you two examples. This one is very modern. This one is very old. So who of you have ever heard of Brodmann areas? Great. So Brodmann was an anatomist from way, way back. I think this atlas that he created was published in 1905 or something like that, which is really the golden age. People started to understand that the brain was a specific type of tissue that you could stain, and then you can look at subdivisions. So what we see here is based on cytoarchitectonics. That is to say, if you look into the tissue after staining, you will see that the histology at the microscopic level between cells differs from brain region to brain region. And of course, then you can say, but here is where this cell type starts to change into another cell type when I move in this direction. So there is a boundary here. Yeah? And so you can parcellate different regions of the brain based on these low-level, very basic biological, histological properties. We call this a cytoarchitectonic atlas. And it's old, people don't use it so much anymore, but specific regions can still be designated by this BA and then a number. That always stands for Brodmann area, whatever. So for example, BA17 is this first visual region. That's why I know it. All the others, I don't know. Yeah. So of course, you don't have this on the first subject level because you need to kill someone to get this information. So you could have it on the single subject level, but you cannot do any functional MRI afterwards. Yeah, so that's a pity. So you get very beautiful, high resolution information, but it's going to be an atlas that you're going to have to use across many subjects, right? There's no personalized information there. Now, this is from 1905. There is now a big brain project from Jülich, the city in Germany, where people are really doing this at the fine scale, and they're really looking at gene expression. So this has really spawned a huge sort of data set that is now starting to come online. So you can actually look at this type of information at, I think, something like 50 micron resolution. You can go to their website and really zoom through this, this brain. Not important for functional imaging, but I just wanted to say a bit about that. So another thing is that you can do it in the living brain is that you can use multiple MRI images and get something that can be a proxy of histology. So if you look at what people like very high resolution anatomical images for, it is often this, that if you take multiple contrast images, we've spoken about this T1 weighted anatomy. We've also discussed T2 star weighted images. You have many more of these images. And if you get something like 4 or 5 images, you can actually discern what types of cells live in a certain voxel because you expect high T1, low T2 star, maybe a medium MP. So you have a bunch of these acronyms. All of them scan something differently, but you all get values in every voxel. Yeah? So you can actually start to classify tissue types based on these MR images. So then the idea is that you can do something very similar to cytoarchitectonics, but you're now doing it in a living brain because you don't have to kill someone. Now, one of the things that people can do is take a T1 weighted scan and divide it by a T2 weighted scan, a very simple operation. And people say, so this is still sort of out in the open, but this definitely is some proxy for myelin content. So who of you know what myelin is, and who of you do not know what myelin is? If you do not know what myelin is, speak now. Yes, great. So myelin is a very fatty protein that surrounds the axons of neurons, and its function is to facilitate the communication by those axons. The effect of having myelin around an axon of a neuron is that the action potentials that the neuron fires are going to be propagating something around 10 times faster than the cell would be able to communicate if it didn't have the myelin. Yeah. The thing is, it takes a lot of energy to set up this myelin structure around the axons. Yeah? So there's this trade-off that the brain is always pursuing. Yeah? That is, do I invest building all this myelin for this brain region, right? Because you may have to break the myelin down if the function changes, right? So the myelin sort of sets the function of a brain area in stone. Like if you're building a building, the foundation, right? That's really important because you can't change it afterwards. So myelin is something akin to that. So if you have a brain region that has to perform a lot of different functions and have a lot of connections with different areas, that's not going to be very myelinated. Okay? But if you have a brain region that is very fixed in terms of its function, right, then you can invest, because then it makes sense. You're always going to have to do the specific thing. And then it starts to make sense to really, put in the energy and start to build that infrastructure so that you can be really good at that single thing. That's what the myelin allows you to do. Now, with that in the back of our minds, we can look at the brain and have a little look. Who of you know a bit about anatomy? I'll go into that next week. So can anyone tell me where these high values, so high here is red, and that means more myelin. Can anyone tell me where those regions are in the brain? Yeah.
Motor cortex.
You said primary sensory, I think, and he said motor. Great. Excellent answer. Well done. So, primary motor cortex. It's like, am I moving this finger, right? Primary auditory cortex is that sound that I'm hearing. Primary visual cortex is that specific pattern in vision, right? So these are where the incoming information comes in. It's always in the same format. We've discussed this, right? The retinotopy is very fixed. It's a fixed structure. It's due to the projection of the image onto the retina. And similarly, for the auditory cortex, you have a cochlea. It will just pick up frequencies of sounds. You're stuck with that, right? And the same for the motor cortex. If a neuron here fires, you're just going to move your pinky, right? You're not going to move anything else. So this fixed relationship of these primary regions, the fixed function, allows the brain to start to really invest in its architecture there. And so if you can really make that really fast, you actually let the rest of the brain be more flexible. So that's what you see here. And we can now really neatly partition the brain, of course, right? Because I don't know how clear this is, but there is actually a pretty strong boundary here between V1, the first visual region, and V2. It only made sense for the brain to really really strongly invest in V1, and then V2 is just the second stage, so it can be slower. Right? So you really see a very strong sort of step-like gradient in terms of this myelin content. You can just delineate in a single subject, you can delineate V1 based on this type of MRI information. Right? So this parcellation you can do based on anatomy, right? Especially if you have anatomical scans that are informative concerning histology. But that's not the only thing you can do. You can also look at just more basic anatomy. You can just look at the thickness of the cortex. Yes. The what? The white matter, no, so this was all gray matter. So it's the myelin content between the cell bodies. So inside one of these sensory regions, all of the cells need to talk to one another to fine tune their behavior. And so that processing is also myelinated, yes. No, good point, good point. That was gray matter, yeah. So we can also look at the thickness of cortex. It turns out that, for example, if you want to know the difference between where the motor cortex is and where the somatosensory cortex is, they're right next to one another, and I'll explain more next week in the neuroscience lecture. But you see this very, very beautiful, strong line between two regions. That's the difference between motor and somatosensory cortex. Motor cortex is really thick. Somatosensory cortex is relatively thin. Also, V1, you won't hardly see it here because it's so thin. So you notice that you have all sorts of extra information just from the anatomy that allows you to say, hey, this is this brain region, this is that brain region. And so based on all of these types of information, what we can do is create these sort of atlases that really delineate regions. And we're going to use things like myelin, we can use things like curvature, we can use things like thickness, all of these different things we can use. And this is just based on anatomy. We can also then just do a bunch of MRI tasks, fMRI, find blobs, and use that type of information also. Yeah? So you can actually supplement all of these different types of information to be able to parcellate the brain. This also means that the more information you have, the less you need to depend on an atlas. Right? We've talked a bit about that this morning also, that an atlas is never perfect, right? You cannot distill the properties of an individual from something that you would derived previously from a whole population. People are unique and you'll lose information. So the more information you have on the single individual, the better you know how their brain hangs together, what the different regions are in their brains. So you can use task fMRI for this. So now that we have a beautiful parcellation of the gray matter, shelve that idea. We now have a beautiful parcellation of the gray matter. Okay? I'll just go and add a few ingredients and then we can get to how we use these parcellations. Okay? So this other type of information that we can gather from the brain is this tractography type of information, right? If we really want to look at the connections, and now I am talking about white matter. If we really want to talk about the connections, knowing what connects to what, right? And so the old work that was done in this field is all horrific Frankenstein material because they used animals, monkeys predominantly, and used these tracer studies. So what you do is you take a virus, for example, a rabies virus that we know can travel from cell body to axon to dendrite to next axon. So you can actually trace where, so the virus will take the path along the neurons connections, right? You can do that, inject a virus, label it with some staining technique, inject it locally in some portion of the brain, and then you can wait. You wait for 1 day, and then you have this sort of speedometer that you know from the literature, and you say, oh, if I wait 1 day, it's going to be 1 or 2 synapses away, right? So I can check. If I sacrifice this animal after 1 day, I will be looking at the connections from where I injected 1 synapse away. But if I wait 2 days, it's 2 synapses. If I wait 3 days, it's 3 synapses. You get the point, but the shitty thing is now you need 3 animals, right? So if you want to make an entire time course for different regions, you need a zoo of animals. This is what people did in the 50s and 60s. People don't do it anymore, but it's quite hideous, this type of experimentation. So what you end up seeing is, of course, you have an injection site, and then as you look at day over day, you see this labeling fan out, right? Of course, this distributes as you go from synapse to synapse. But this allows you to trace this network of connections in the living brain in a very, very specific sense. Okay? There's 2 ways of doing this. One is a retrograde, depends on the specific type of virus that you use. You can do a retrograde tractography in this way, which goes from the dendrite to the axon, so opposite to the direction of information transmission, which is from the axon to the dendrite. And you have anterograde, which goes from the axon to the dendrite, right? So it follows the same direction as the information flow between the neurons. So you've already labeled the virus, you stain the tissues, and you find out where these connections end up. And I want you to realize how gruesome research was in the 50s and 60s, because this is not the only thing where people really didn't care about animal well-being or anything like that, right? And it's quite amazing to see this sort of work where you have papers, people reporting 120 monkeys tractography. So it's really insane. Yeah, what can you say? Like old white dudes, right? So, yeah. Why is the cingulate cortex function so flexible? Sorry, I missed that. So that's a question. That's a good question. I have no idea. Cingulate cortex is, is this band around the corpus callosum. People think it's part of the older parts of the cortex. Why is it so flexible? So different parts of the cingulate cortex have different roles. This frontal part of the cingulate cortex, oh, this is, by the way, a task fMRI, so I should go maybe here. Yeah, so you see that there's different parts of the cingulate cortex. It actually runs from here to here. Why is it so flexible? Yeah, so you'd think that this part is really flexible and this part is really inflexible from this myelin map. Trying to answer your question. So the frontal part of the brain is responsible for behaviors, for actions. And this part of the brain, the anterior cingulate cortex, which is part of the frontal lobe, it does things like error monitoring, like, am I doing the right thing? And it will start to fire. There's neurons there that are sensitive to things like response conflict. Should I get, this sort of wondering what to do sort of thing, or experiencing that the choices that you're making are becoming harder and harder. That sort of thing lights this up. And you can imagine that sort of thing needs flexible types of operation, whereas this posterior part of the cingulate is much more sensory oriented in terms of its processing. I hope that answers your question somewhat. Any other questions up until now? No? No. Great. So luckily, we don't have to do tractography like this anymore. And luckily we have this MRI machine. It's incalculable how many animals were saved by the advent of fMRI or MRI machines in general. Because in this case, we can also do tractography with the scanner, right? And this is mostly a reminder. I think I mentioned DTI before, did I? I forget. Yes, right? Yes, so, and this is just a reminder, right? The scanner really can't do anything. Well, it can't fly and that sort of thing, but depending on the program that we run on the scanner, the sequence that the scanner performs, we can not only focus on different histological properties of the tissue, or scan really fast and then look at blood responses like we do in fMRI. You can also look at the diffusivity of water. So that is, how does water diffuse and in what directions does water diffuse in a certain voxel? So what you do is you label water at T1, at time point 1. So that's not T1 for contrast, but time point 1. And then you can sort of check whether it's moved in a certain direction at time point 2. So you look at a difference, and you look at a difference in certain directions. Right? So you can imagine that if water is able to move freely, it diffuses much faster, and thereby you get a stronger drop in this label that you've performed. Yeah? But this would be in a bucket of water, where diffusion happens in all directions at the same rate. So you just get this sort of normal distribution and diffuses away. But your brain, of course, especially the white matter, you have a lot of structure. So the white matter has all these axon bundles, and what they do is they restrict this diffusivity of the water, right? The water can very easily diffuse in the direction of the axon, but it cannot, go through the membrane of the cells very quickly. So if you have an oriented bundle of axons, what you'll notice is that the diffusivity of the water in that bundle will not be homogeneous. It will not be isotropic. What will happen is the diffusion will be constrained into a single direction, back and forth along the directions of the axons. Yes. So that looks something like this. So you can do all sorts of data analysis on this. If your bundles are oriented like this, you end up with this diffusivity measure like so. What you do is you measure this diffusivity in all sorts of directions. So you get single values in every voxel for a bunch of directions. And then you can start to analyze this. And for example, say, this diffusivity is elongated in this direction. So that means that the majority of the axons inside my voxel are oriented like this. Yeah. So what that means is you can sort of, and so this requires rather complicated mathematical modeling because it's all geometry and that sort of thing. But you end up having like these models of a diffusion that's anisotropic in different directions. And what comes out is a couple of different important measures. So the first is you get this fractional anisotropy, right? Like I said, isotropic diffusion will give you a ball, like a basketball sort of thing, a globe. Isotropic then means equal in all directions, right? Like we said about voxels, they can be isotropic, meaning that they're the same size in all directions. Anisotropy means the opposite of this. So cigar shaped in this case. Yeah. And fractional just means how much, like a relative ratio. And so what you get is really the cigarness of the diffusivity at every location in the brain. And so this allows you to map out the white matter and map out how strongly the bundles in the white matter are oriented all in the same way. Yeah. So in this case, we don't really care about the direction of the bundles. We just care about how strong they are. Yeah, because that will change this diffusivity thing. I'm not going to go into the math here, but you have this thing in water. It's a ball, so it's very isotropic. And the more cigar-shaped this is, the higher this value of fractional anisotropy, and you can map it in this way. Yeah. So, for example, in MS, right, it's multiple sclerosis, there is damage to the white matter. This type of scan helps you track the disease progression. Yeah. But of course, we can do this. We do this quantification for multiple directions. So we don't just get this fractional anisotropy. We also get information as to the direction of the cigar, right? How is the cigar oriented? And that, of course, is even better. Like that is much, much better. So that gives you the local orientation of the bundles, not just how strong they are. Yeah. And so based on that type of information, we can actually do tractography. We can try to trace paths from one region of the brain. Remember, I was talking about these parcellations. We can look at how one portion of the brain, perhaps here in frontal cortex, might be connected to some portion here in parietal cortex, because we can trace these paths. Right? And I think, did I show you this? It's one of my favorite like DTI little movie things. Even sound. Like a meditative type of thing. This. So here you see the results, right? This reconstructed in 3D, where we're actually tracing the fiber bundles. You have a specific directional thing, like up is blue, they're pointing up. The Y direction is green, and the X direction is red. So everything going sideways is red. Everything going front back is Y, is green, and then top to bottom is blue. Yeah? So you actually have like a sort of, yeah, you can actually look through this and really appreciate where all the bundles are going. Huh? Great. Connections, awesome. So what can you do with this? Like now that we have information, what are we going to do with it? Well, you can start to check between regions how much they are coupled. So you can start to quantify the connections between brain regions, right? So first, having parcellated these, you can look what is the strength of the connection between this parcel and another parcel? And you can do this for all combinations, and that can give you a matrix like this. And so, for example, you can then see how this deteriorates when you age, when people are diagnosed with multiple sclerosis. So you get a very sort of common format for your data, right? And this can be very insightful. Now, when you have a matrix like this, what you can do is specify a cutoff value. So you say, I'm just going to say that anything below a certain value is not connected, and everything that is above a certain value of this connectivity, it is connected. And that allows you to then set up a graph network for the brain. People do a lot of analyses, like graph theoretic analyses on this sort of network structure. Now people like Facebook do a lot of this type of modeling, graph theory modeling on social networks. And in this sense, the brain isn't much different if you've already gotten to this format, right? And then you have people making these beautiful, beautiful figures of how to present this type of connectivity. So you're going from, what is this? I think this is like frontal cortex on the right, frontal cortex on the left, and then you go to like sensory cortex. So you see how everything is connected in the brain, right? I generally tend to think this is not the clearest of all representations, but yeah. Okay, the text really doesn't matter. Because I already said it. Yeah, so let's have a break. And then I'll talk more about functional connectivity, right? So the left half of that initial diagram we've taken care of, right? There is certain fixed properties that we can relate to how brain regions are connected, and we can parcellate them. Now, functional. I mean, after the break, functional. Jeez Louise, I didn't. Yeah, no, you're right. Okay. Okay, I'll try to splice in anything that was lost in the recording. My apologies. But this is, I'm not using that, to be clear. I'm not spying on you. Yeah. Great. Yeah, so I'll try to splice in some comments from this. So, I want to highlight one other type of analysis that people do in things like this, in experiments like this. And it's this type of analysis that you can do regardless of whether you're doing a resting state experiment or anything else. The idea here, and it's called ICA. Who of you have heard of ICA? Great. So it's a relatively standard signal processing type analysis technique. The idea is that when you're doing a recording, such as you're doing an fMRI, what you're actually doing is you're recording from all of these voxels, and each voxel, it gets some signal from, let's say, task. It also picks up some signal that's related to machine noise. You get really weird types of things like arousal-based fluctuations. Remember in that first lecture about the bold response, I showed you these monkey results with electrophysiological responses and then the bold response and some sort of expectation signal that's really weird. That sort of thing you're measuring. And it's also in your voxels, right? So there's going to be some component of these weird arousal related things that you're also picking up in each and every voxel. And then there's things like pulsation artifacts. This morning we looked at how the brainstem has these very, very strong pulsation artifacts, right? So when you're recording from a single voxel, there's some mixture of all of these signal sources that you're picking up on. Right? It's never just one. There's always some thermal noise, some physiological noise in there. The idea is that these noise sources, they are actually, for many voxels, more or less the same. There's structure to this noise. So a lot of this variability, for example, related to pulsation, two neighboring voxels will share it, but not really just that. Also, any two voxels that are near a blood vessel anywhere close to the base of the brain will be pulsating in more or less the same way. Okay? And that is that what that means is that all of the voxels that you're sampling from there will actually be showing a time course very similar to this. Okay? And the same goes for if you're doing a task experiment. All of the voxels that are sensitive to this task will more or less do similar things. So signal sources are shared across voxels, and any single voxel picks up many different sources of signal variation. Yeah. Now, this is something that we are totally not looking at when we're doing GLM, right? The single voxel analysis, because we only care about the single voxel. But this is about the different types of signals that voxels share. Yeah. So what you can do is, it's called independent component analysis. I'm not going to explain the maths behind this, but the idea is what this technique does is it separates spatiotemporal sources of variation across the brain. Yeah. So what it will do is it's a totally data-driven technique. What you will do is say, here ICA is my fMRI data set. And it will start running and it will produce components. Now, any component has a time course and it has a spatial pattern throughout the brain. So when you do ICA on an fMRI signal, you end up with very stereotypical components. So some of them are going to have a structure like this in time and a spatial structure that shows you the arteries in the back of in the base of the head, yeah, because it's pulsation. And pulsation related signals are very similar, and ICA will find this similarity between those signals from those different voxels and pick it up as a separate component. Yeah? And similarly for tasks, similarly for different types of noise. And so what ICA allows you to do is really partition out different parts of the brain, really find specific networks that are doing specific things. And here I'm showing two, right? Their spatial map is here and their time course is here. Again, a spatial map is here and a time course is here. Usually when you do this sort of analysis, you get out, let's say you have a run of 300 time points, you can get out like 100 different components. But what you see is, so these are both reasonable time courses, as in this might be related to an experiment. And indeed, you see and you look at, these are actually regions of the brain that we know are involved in certain types of tasks, right? So it's picking up functional information from the brain. And similarly here, we get a similar thing. Right? So important is that what we get out is a spatial pattern and a temporal pattern. And that's one of those components that contributes to the signal that we measure in each and every voxel. Right? So you'll be able to find the signature of this component in any voxel in these regions. Yeah? Now, specifically, this ICA performs a statistical procedure where it tries to separate out these components, where it tries to, what it does is it tries to make these components maximally independent of one another. There's different ways in which you can do this. And ICA is one that works relatively well with this type of brain data. This one? So this is, something that you could convert into percent signal change. So, yes, yeah. And so what people usually do, I don't know how many of you are familiar with this, but this is a frequency spectrum. Also something we'll go into in the third notebook. So frequency spectrum will also tell you what is in the signal, right? So for example, this heartbeat thing, it happens about once a second, right? So you expect a lot of signal around 1 hertz. And because of the HRF being so slow, we don't expect any functional activations at 1 hertz. Right? So anything that has a peak around 1 hertz, you can definitely say, this is likely going to be a pulsation related artifact. Right? Similarly, breathing related artifacts always happen at around 0.3 hertz because that's your breathing frequency. And so you really can interpret this structure, and you'll go into this a bit in the notebook, so I won't go into it much more. But so these are signatures that you can also attribute to these components. Yes, what is pulsation? It's the beating of your heart and the fact that your vasculature is flexible. And so what that means is that when your heart beats, there's actually a wave of blood going through your entire body, right? Because otherwise you would not live. But this is pretty strong in your head because your head rests right on top of your aorta, right? This the aorta arc that leads directly into the carotid artery. So you get a lot of pulsation in the bottom of the brain. So pulsation really is the bobbing of the brain, and specifically its basic vasculature on the heartbeat. Yeah. Great. So what you can also imagine, and this is a side point, but if you are able to pick up cardiac related, so pulsation related artifacts from an ICA analysis, what you can also do is just say, then now I'm going to delete this component from my data. So people also use it to denoise their data. That's actually something that people do a lot in EEG too. So they just check what looks like a blink in someone's data, and then they just say, any ICA component that looks like a blink effect, I'm just going to throw out of the data. Yeah? Now, this logic of denoising data based on this type of analysis is really what the Notebook 3 is all about. Okay, now, having done all of this, right? Now that you have an idea of, we can pinpoint the voxel using seed-based connectivity to find how things are related to one another, right? We can use these parcellations that I spoke of in the first part of the lecture and really look at all-to-all like connections between all of these patches and do like a network analysis, a graph analysis. Or we can do something like ICA and thereby get these components and also pick up which brain regions co-fluctuate. And that's all that we wanted to look at, right? How do things co-fluctuate? Now, when you do this, you find standard things. I tried to point out this sort of standard thing in the movie of the ongoing resting state, and I was horrible at it, for which I deeply apologize. But if you aggregate data, you are able to look at the structure of these resting state scans. And it turns out that there are specific networks that people always find that co-fluctuate, and that really tell us something about this endogenous intrinsic co-fluctuations that the brain goes through when people are, whatever it is that they're doing when they're in resting state scans. Yeah. So I hope that you have an idea of all of the caveats, not being able to know what is signal, not being able to know what is noise, et cetera, et cetera. But the beauty is you do get these very consistent networks out. So let me just highlight. So the self-referential network is when you are thinking about personal relationships. When you're performing things like empathy tasks or theory of mind, like what should I do whenever this and this is the case? This core network has to do with trying to feel your body. Like if you're in a yoga class and you're focusing on your breathing, it's this type of activation that you see in older parts of the brain, like the insula. And I'll talk more about that next week when I go through the brain and tell you a bit about all the different brain regions. There's an attention network, ventral attention network, that picks up things that happen suddenly in the outside world. So for example, you might suddenly notice something, that a noise that the scanner is making while you're in this resting state scan, and that will co-activate all of these regions. And then there's a dorsal attention network that is your endogenous direction of attention to the outside world, like oh, what's that on the corner of the screen? I'm going to, think about that for a bit. That sort of thing. This network. Yeah. Then there's like, maybe you're twisting your fingers a bit without knowing. You get all of this sensory motor activations. You might suddenly have an inner voice or think about something, again, like a sound that the scanner is doing, but you're doing so in a more prolonged way, focusing on auditory attention processing. Then there's visual cortex. You just, I'm looking, something like that. You're blinking, that will definitely show you visual activations. And then there's this default mode network, which is really interesting. And that is when you're just thinking, that really is daydreaming. So this default mode network is very interesting, or it's thought to be very interesting, because it really has this sort of back and forth between, for example, the visual system. Whenever you think of, or you're busy doing something on things that happen in the outside world, you're using vision or audition, or maybe your touch. Yeah. On the other hand, sometimes you're just thinking about stuff, about life in general, really daydreaming, right? And it's really coming from memory. Then you get these regions activating. Right? So this daydreaming, if you, for example, look up daydreaming and relate it to where in the brain this is that region. And what's interesting is this default mode network is actually the part of the set of regions in the brain that have grown most in humans relative to our ancestors. And so this sort of social thought or like planning, all of that sort of thing, much of that is happening in the default mode network. So we do learn something about the global structure of the brain when we do this resting state type of analysis. I'll talk more about brain regions and what they are responsible for next week. So on Friday, I'll be, it's going to be a relatively brief lecture, most probably. What I'll do is I'll go through the different projects in the project notebook. Feel free to anticipate and do that yourself. But what I'm going to do is go through these all three projects. I might show you a bit of the underlying literature for each of the different projects, and show you like a lot of the basic analyses have already been done for each of the three projects, and you will be able to then work from that. So I'll show you those basic analyses. Any questions about the course and how we're going to continue, and any other? Yeah? No, I'm working on that with Nikos, who's also going to give the MR Physics lecture. So yeah, we have a rough version and we're working on ironing out all of the creases. Yes, so I want to highlight about that fourth thing. It's rather separate from the projects. It's also not as intense as, for example, the first and the third notebook. And it's really to give you an idea of, where do these images come from? Yeah, I definitely want to highlight that it shouldn't get in the way of your being able to work on the projects. Yeah. Yes. Yeah, I get that. Yeah. Yes, there will. I'll put those online as soon as possible. Cool. Yes. No. There's a secretary that is sort of the guardian of the exam commission, and they wanted to know whether I couldn't reschedule where the exam is being held in the first place. It's something I'm struggling with right now, but it's ongoing. Now, I'm very sorry that this is such a conundrum. I understand that that's really frustrating. Yeah, sorry about that. Yes. Upload? Oh, like so. Yeah, so I was gonna say like around exam time, but if you feel like you want to work on it after the exam also, that's also fine. You notice I'm not really into deadlines. Next year somewhere. Yeah, whatever. Yeah. Yeah, there will be a practice exam. I'll upload it on Canvas. Yeah. Oh yes, and so how we distinguish these different resting state networks is not from the resting state experiment itself, but rather from the relation of these regions in other experiments. So we know that the visual network is visual because we've done visual experiments other than the resting state experiments. Yeah? So if you ask someone to think specifically about their personal relations for a couple of seconds, then you can map out this default mode network or self-referential network, things like that. So it's referenced against, no, definitely. There's a lot of assumptions in there. And so this is, a part of the vagueness that goes with resting state in general. There's also a conceptual vagueness, like what are we talking about conceptually? Okay, I'm gonna stop sharing. And oh, there's my flower pot. And stop the recording.